Okay, so now we're going to talk about actually filtering the sample and concentrating it and getting it ready to send over to the lab. So you have a filter apparatus here, and you also have the M-Vac in its holder, and as you can see, it still has the sampling bottle on it, and it's now full of the solution and the DNA material. So the first thing you want to do is take this sample bottle off, and you do that by grabbing this ring and simply twisting clockwise. Twist the bottle clockwise until it falls off, and make sure that once it lets go that you don't spill that sample. So now you have the sample there, and then you have your filter apparatus next to it. What you want to do now is take the vacuum tubing off of the M-Vac. So you just work it back and forth until it comes off of that, that notch right there, and then you insert the vacuum tube, and you can pull this solution line off a little bit. You insert the vacuum tube right there over the top of the port on the solution, or I'm sorry, the filter apparatus. Pull the cap off, and then turn the vacuum pump on. So once you've turned the vacuum pump on and you've wet the filter, you take your sample, swirl it around a bit to make sure and suspend all the DNA material in that liquid, and then slowly pour it into the center of the filter. And if it's a clean sample, it should filter right through. Swirl it a little bit more, pour, swirl a little more. So if you're concerned at all about any DNA material still being in the bottle, you can take the sampling head and spray it directly into the bottle. Get 10 or 15 mils in there. Swirl it around. Get up there on the sides, and again pour it through there. And if as you were pouring, and it poured rather quickly, and it took a little bit of time to filter through, you can take the sampling head and just spray the sides of the cup so that it washes down any DNA material that may be up there. But other than that, we're ready. So you turn the vacuum off. Lastly, you notice here there's still pressure on the vacuum pressure on the filter. So the way you want to do this is to just gradually release the pressure. You can either do that just by leaving it alone for a while, or if you're impatient like I am, you can just gradually work this tube off, and you'll hear the air let in there, and now it's ready to be released. Again, there shouldn't be anything inside the filtrate down here. All the DNA should be on the filter, which is inside the cup. So again, once that dries, they'll be ready to process it.